Once upon a time, there was a boy with orange sclera that got made fun of. Recently, from the goat bio teacher, Ms. Pabon, we learned about meiosis. Understanding that it causes a variety in offspring, he wanted to go back in time to change the color of his sclera, so he contacted his good friends Marty McFly and Dr. Mendel to go back in time to retry his chances with traits life. Hello, and welcome to our meiosis video project, narrated and illustrated by Arlen Laperio, produced by Ritlak Badwarden, and conceptualized by Akshay Jagd. You guys have just met our main character Jeff with orange sclera, and you may need to understand the full process of meiosis to understand how he got this trait. But before we can even begin talking about meiosis, we have to learn about a process the cell undergoes called the interphase. This consists of three main parts. The first part is G1, which is when the cell begins to grow and expand. You can see that the cell actually grows from being pretty small to being pretty big. The genetic information of the cell is still very organized. But as we move on to the second part of the interphase, which is called S, notice how the DNA inside the nuclear membrane gets messed up. This is because in S, the cell replicates its DNA, and you can tell that S has occurred because the chromosomes are now duplicated, resulting in an almost X-like shape when the genetic information condenses into chromosomes. The nuclear membrane also begins to disassemble at this point. While this is happening, you can see that another centrosome, which you can see by the black shape around the nuclear membrane, has been replicated, and it's making its way away from the original centrosome. And the last and final part of interphase is G2, where the cell has its final preparations for cell division. The cell that we are looking at is a 2n=6 cell, which means that the chromosomes are diploids and there are 6 chromatids. Now we move on to meiosis, and the first part of meiosis, prophase 1. Prophase 1 is where the sister chromatids form homologous pairs of chromosomes, and to pair these homologous pairs of chromosomes, they are connected via centromere. Notice that the centrosome has now moved a little bit further away from the original, and also notice that the nuclear membrane is fully gone now. And also, in prophase 1, there is an action that takes place called crossing over.
I miss my cocoa butter kisses, hope you smile when you listen.
Where genetic material is transferred between homologous chromosomes, which increases genetic diversity and variation. Now comes metaphase 1, which is where the chromatids line up at the middle of the cell at a place known as the metaphase plate, with the homologous chromosomes facing the now opposite centrosomes. And there are many ways that the chromosomes can orient themselves at the metaphase plate, with each combination changing how the resulting offspring would look. The next part of meiosis is anaphase 1, which is when the homologous chromosomes split apart from each other and go to the opposite centrosomes in the cell, with the help of each centrosome's spindle fibers, which in our drawing and diagram is indicated by the black lines going through. Lastly, in the process of meiosis 1 comes telophase 1, which is when the chromosomes arrive at the opposite centrosomes and the nuclear membrane is formed around each set of chromosomes. Then cytokinesis occurs when the cell divides, as indicated by the red line, into 2 daughter cells, which are each haploid cells, or otherwise n=3. After meiosis 1, which is PMAT1, comes another set of PMAT, also known as meiosis 2, and the first part of that is prophase 2, which is when the new cells, the new haploid cells, have a small, almost interphase-like stage, but they do not replicate their DNA. And you can also see that the DNA of each cell becomes messed up again, and another centrosome is created in each cell. Next is metaphase 2, which is when the chromosomes align at the metaphase plate, the middle part of each haploid cell. Now, in anaphase 2, the sister chromatids split apart and move towards the opposite centrosomes. Finally, in this video comes the last step, or rather 2 steps, known as telophase 2 and cytokinesis. And telophase 2 is when the chromosomes finally arrive at the centrosomes, a nuclear membrane is formed around each set of chromosomes, and then another round of cytokinesis occurs, and now we have 4 daughter cells that are each haploid and n=3 cells, and they are all completely genetically unique. As you guys just saw in the video, we start with 1 cell and ended with 4. And to be more specific, we started with 1 diploid 2n=6 cell, and we end up with 4 haploid n=3 cells. This is all thanks to meiosis. Now, moving on to the bigger picture outside of meiosis, you may be wondering, how does meiosis actually relate to Mendel's 5 claims? So Mendel's first claim is alternate versions of genes account for variation in traits. And while meiosis itself is an expression of genetic variation, the process introduces combinations of genes, which showcases the alternate versions or alleles in the gametes. Mendel's second claim is that for each trait, you inherit an allele from each parent. And in meiosis, the initial cell inherits 2 copies of each gene, one from each parent. and it transforms from a diploid to a haploid cell. And through meiosis, these cells actually become haploid gametes. And as we know, when two gametes, sperm cells and egg cells, combine during fertilization, the diploid state is restored, showing how one will inherit an allele from each parent. Moving on to Mendel's third claim, the law of dominance. In meiosis, we see that each gamete only receives one allele for a particular trait, and for example, if this allele is dominant, then that will show, but if it's recessive, then it may be overpowered by a dominant, thus proving our law of dominance. So Mendel's fourth claim is the law of segregation, and actually, during the cytokinesis part of meiosis, this is actually the most observed law, because the two alleles for a character are separated into different gametes after telophase, and the cell has to divide into two smaller cells. Now finally, moving on to Mendel's fifth and final claim, the law of independent assortment. If we go back and recall to metaphase in meiosis 1, the chromosomes align along the metaphase plate, which, as you may remember, is the middle of the cell. And because there are so many chromosomes, in our case there was only 3, but in most mammals, like the human, there are 23. We actually witness the law of independent assortment in action, because the way that the alleles for different traits are sorting independently creates millions and millions of possible combinations for how an offspring may look. After watching the full process of meiosis, Jeff realizes why he has a different color sclera, and when he tries to change it, a complete resets who he is as a person, and he is warped back to the future, now with a fully deformed face. Thank you all for listening and watching our video. We appreciate it greatly. Happy holidays.